Well, fantastic. Welcome everyone to this new year. And I'm very excited. My name is Amanda Brown. I'm an associate professor here in the Department of Neurology at Johns Hopkins University. And I'm really delighted to introduce our speaker today, Dr. Carlo Colantuoni. He's an assistant professor of neurology and neuroscience here and an adjunct at the Institute of Genomic Science at the University of Maryland. He pursued his undergraduate studies at Princeton before his PhD in neurosciences here at Johns Hopkins, and then did postdoctoral fellowships both at the National Institutes of Mental Health with Joel Kleinman and at our School of Public Health with Scott Zeiger. He started his first faculty position at the Lieber Institute for Brain Development, collaborating closely with Dr. Ron McKay's brain development group, and then also joined our Department of Neurology here at Johns Hopkins at that time. His group works to elucidate conserved molecular mechanisms in human cell biology and pathology by developing and applying novel data decomposition tools, curated collections of multi-omics data sets focus on particular cell and disease contexts. His lab integrates both in vivo and in vitro stem cell systems to gain deeper understanding of human functional genomics, focusing first on human variation across early embryonic and neocortical development, and more recently on neuroinflammation and neurodegeneration. He's worked extensively creating the neuroscience multi-omic, or for short, NEMO, analytics platform with collaborators at the University of Maryland. And this has led to the neocortical development data exploration environment and implementation of projection tools for delineating shared molecular dynamics across biological systems. And in addition, I've had the pleasure of working with Dr. Colantuoni over many years here at Hopkins, as he has served as an outstanding mentor and trainer for students from the high school level all the way to early career faculty. So thank you so much, Carlo. Please
Thank you,
take it away.
Amanda. Thank you so much. So I'm hoping today to not only show you what can be done with the public data resources out there, but enable you hopefully to do some of it yourself. So I added like a word, one extra word or a couple extra words to the title, because in general, people think, and often it's true, that you can't go use all of this multi-omics data that's out there, even if it's relevant to your cellular system of interest, if you don't have either coding skills of your own or a bioinformatician to do this. So I'm going to show you the work I do to empower my own research, but everything I build to do my own research, I build with the idea in mind that anybody should be able to pick up this tool and use it that I just used to find something. They should be able to pick it up and use it on either the same data or more data to really fuel discovery. So, and please do reach out after and interrupt me in the middle of this talk as I go through, so we can make sure we stay on the same page, and I can hopefully show you how to use these tools that really I'll explain as I go on. There, each one is sort of the math tools, and then the online tools for you to surf data are each sort of grand scale collaborations with lots of people involved. So we really want to make these tools available to people. So the underlying idea here is that there is data out there that you are interested in that you may not be using today. No matter what field of biomedicine you're currently in, what you're studying has been studied with RNA sequencing, with epigenetic sequencing, and at different levels, different species and model systems. The in vitro systems have exploded as incredibly powerful tools to study everything in biomedicine. We really couldn't study functioning living human cells across the plethora of cell types until now. We can study anybody's cells in any context now. It's kind of overwhelming, and all these different data types are coming in. But it is not at all the case that we have made these data easily usable by people who don't code for a living and use large amounts of data. So we are trying to bring some of this data together for focused areas. We can't do this for all the data in the world. So the data is out there and sprinkled for these contexts that Amanda mentioned and I have here mentioned on the bottom. We have gone and brought as much of the public data as we can together to focus on that particular disease context. As Amanda mentioned, I've done a lot of cortical development and very early embryonic development models. But today, all the examples and data analysis and data we look at will be in neuroinflammatory, neurodegenerative contexts to show you how this works. And again, at any level that this can be useful to you, either talk to me now or talk to me later. But I'll try to show you how it can be useful at multiple levels. So step one is to get the data together. Step two is what we do with it, of course. And these are sort of words that may not mean much to you right now. And as we walk through today, hopefully it'll make more sense to you. So decomposition and projection are two steps of data analysis once you have data in hand. The papers I have here are from long-standing collaborations with Brian Caffo at the School of Public Health and Biostatistics. We've had a really long, fruitful collaboration that now also includes Genevieve Stein O'Brien, who was my first graduate student and who is now faculty in the neuroscience department. So we're still working together to build these tools. But the basic two steps we're thinking about here, decomposition and projection, are decomposition is breaking apart. So if you think of each paper you know of that has a data set of interest, think of that like a matrix or a spreadsheet. And maybe there's 10 data sets you know out there that are relevant to your cellular question in mind. Each one is a matrix of, on the rows, genes, and on the columns, samples. Now that can be very many different things. That can be transcriptomic data, so you know how much of each gene is being expressed and how much in each of your samples. It could be a small data set of 5 bulk RNA sequencing samples versus 5 other treated bulk RNA sequencing samples. Or it could be a huge data set with 1,000,000 columns where each column is a single cell, and that column describes how much of each gene that cell is expressing. So the scope and range of these data sets is massive, and we definitely collect tiny experiments and giant ones as well. You know, these atlases give you great sort of assessment of the landscape of the different tissues we're now becoming molecularly intimate with, that we know so much about. But then tiny little studies can be interesting if they have a particular treatment or genetic implementation that's of interest. But either way, coming back to these two words, the decomposition is grabbing one of those big sheets of data and finding patterns in it or pieces in it. The mathy way to talk about it is finding the low dimensional order in high dimensional data. So if you have 30,000 rows, each one is a gene. It takes a long time to look at 30,000 genes. You don't really understand them looking at one at a time. But there could be 5 or 10 patterns that repeat in those genes that are of interest to you. Maybe something that comes on early in a time course with lots of genes and something that comes on later, a different packet of genes. So the decomposition is really a mathematical way to take these data sets, either one at a time or several at a time, and we'll see how to do that a little bit differently, and break them apart into pieces. So once you have that piece, it's a quantitative description of either a epigenetic or transcriptional module. And projection is basically the process of assessing the strength of the module you found in maybe your cell type or treatment of interest and looking at other data sets. When I squirt this cytokine on microglia in a dish, does it turn on some of the same genes that are turned on, say, in vivo in MS when you have an active lesion? And that's really that notion of connecting data sets like that via transcriptional signatures or epigenetic signatures is really what this is about. That's why step one was getting all that data together so that when you get a molecular element of interest from a decomposition, something that you're interested in that a cell is doing, what other cells are doing it in what other contexts? What other agents might push it back the other way or might push it up, where it would, depending on what you might want to do in a cellular system. So those are the two big steps in the general approach I'm going to outline. And really, the third is a way to interface with all this. Now, I myself and many people code in languages like R or Python to do all this. And very quickly, what I realized as I was building my own collection of data and tools offline is that even for my own work, it would be so much smoother and quicker to say, look at gene signature X when I put on a cytokine across 10, 20, 30, or 100 data sets. Doing that with coding gets to be really, you can do anything you want with coding, but if you're doing a lot of things, especially a lot of visualizations, having an automated online system is a huge benefit. So I use the Nemo Analytics site daily for my own exploration, and I build pieces of it daily. And I hope to show you today how it can be useful to you. And this is the publication. It's now in revisions at a journal, but it's on bioRxiv, so you can look at it. This is really where we kind of put together all these collaborations, all the biostatistics collaborations to come up with the matrix math to break apart the matrices and then look at the effects in other matrices are in there. And the tool itself, which this is a screen capture of, is really presented there where you can look at all this data. And this publication is for the brain development environment. And Yash is the first author who really did a lot to really empower this environment. And the NEMO environment is actually housed or built by a group of web developers at the Institute for Genome Sciences across town. Amanda mentioned in my introduction that I've had a long-standing collaboration there and still have an adjunct appointment, so stay connected very closely. And people like Seth Ament and Anup Maharkar over there have really for years been working together. And Rona Herzano, who was there, who is now at the NIH. We've been working together in many different ways on many different projects to really build up the utility and ease of use of this system. Okay, so right now I'm not going to jump to the website, but almost every picture I show you, including these here, are screen captures from the live functioning website. And toward the end, I'll jump on to show you how it works a little more and show you how you might be able to use it. So those are our 3 steps. Get the data together, break it up, and look at those pieces across your data collection, and make sure you can do that in a quick, easy way, whether you're coding or just clicking on the website. Okay. So I have a few slides here that are very much like the recent Grands Rounds I did. And then we're going to dive in a lot deeper again to get into a little bit of the nuts and bolts so you understand how this works and how it might fuel your own work. So starting from a single data set that's relevant to neuroinflammation, this is quite an old study. It's 2012 from Ben Barris's lab. And it was bulk RNA sequencing of sorted cells. So in the mouse, the pink to red is a saline injection versus an LPS injection. So these are peripheral injections followed by central extraction of astrocytes and then RNA sequencing of those bulk astrocytes. So what this is a picture of is the composite expression of genes if you simply compare the vehicle to the LPS treated animals, get a list of differentially expressed genes and take your top 25 genes, and this is their sort of composite expression. So not only do they go up when they treated with LPS, but in this same setup, instead of using LPS, they also did a stroke model, so a middle cerebral artery occlusion, and looked at a 1, 2, and 7-day time course there. So the same packet of 25 genes that gets pushed up in LPS treatment also gets pushed up in the hypoxic condition or the stroke model. And actually these come back down over the course of about a week. So this is an example of decomposition, quite a simple one. You take 2 groups and you do a treatment, you do a differential expression and get a list of genes. So then this is that list of 25 genes. Some of them might look very familiar to people on this call. Lipocalin-2 is a super famous gene in innate immunity. It's has an amazing range of functions, but in one of its most direct is to simply fight over iron with bacteria. So if the bacteria growing in you cannot harness iron, their growth is severely limited. That's one of the many things lipocalin does. It also acts like a cytokine on its own, I think. Um, Another one that's maybe very well known is this SPP1. It's a phosphoprotein that often gets turned on in neuroinflammatory states. And we'll come back to what these genes are, but this is just to kind of demystify it. It's just a list of 25 genes that we observed in Ben Barris's experiment to go up when mice are treated with LPS. Okay. So now, I'll actually send out to the group if I think it'll be possible. Links here. These are the slides, and these are live links. So if you wanted to go see the lipocalin 2 gene expression in this and many other data sets, you can simply click on this. And here you could click on this and see the LPS signature not only in this data set but in many other data sets. And in fact, that's what the next slides are. We're going to walk right through them. We're going to be looking at the aggregate expression of this set of 25 genes. Okay, so, and here, again, we're going to walk through a bunch of other data sets that I've already collected that are relevant to neuroinflammation, and we're going to be looking at this LPS signature in those data sets. So this is from Paul Teaser, who has been a long time investigator of oligos and astrocytes and glia in general. And in this experiment, they took mouse CNS astrocytes and treated with a range of cytokines. They did both bulk RNA sequencing and single cell. And when we look at the expression of our packet of LPS induced genes in astrocytes, we're now in the same cell type. In both cases, bulk sequencing across the top, but of the purified cell type, astrocytes only. And you can see with all the different combinations of cytokine that they play within this paper, all of them turn on this packet of 25 genes that is also turned on by LPS in vivo. And we could look one by one to see which of those 25 are up. This is the aggregate expression, so we don't know which of them is involved more in this increase and which are not. But then you can see the same thing at the single cell level. So this is now a map where every dot is a single cell in this experiment. Again, the treatment of astrocytes with cytokines in vitro. And the ones they treated are in blue, and the ones that were not treated with cytokine are in orange. And you can see, as per the scale bar, all of the astrocytes that were treated with cytokine are blowing up this signature. And there's very few astrocytes that did not see cytokine that have turned on this signature. So this is kind of a proximal experiment. It goes from an LPS treatment in astrocytes to a cytokine treatment in astrocytes, but you could imagine why that would be a highly overlapping transcriptional program and also in the same cell type. But something we know about the innate immune system is that the innate immune system kind of lives in all of our cells. And so now we're going to start to walk through other data sets that work with different data types and cell types to see other places where this is seen. So this is another Ben Barris experiment, single cell sequencing from the mouse CNS during development. And again, so each dot is a single cell from this developmental experiment. And as you'll see right here, there's a pocket of cells that are expressing this packet of LPS responsive genes at higher levels than the others. And this is this light green here. And what the light green turns out to be is postnatal DAM-like microglia. So DAM are disease-associated microglia that were, that DAM signature is derived from Alzheimer's models where they were studying microglia as well. And really, there's a lot of these reactive signatures that are now being seen in the literature and seen over and over. So this is really what we need to do, is get all sorts of data sets together to look at them and see where they're happening and not, and how they can be modified. But where this astrocytic LPS signature is coming up is in microglia now. So we've already jumped cell types. And in fact, they don't have any, I guess because it's early, they didn't have any astrocytes in here, but it's the reactive disease-associated microglia that are expressing this same packet of genes. And again, not surprising given these two cell types and their roles in innate immune responses, but it's interesting to be able to see this quantitatively and across data sets. So now I'm just going to walk through a couple more to give you an idea of what data sets are out there and how we can keep using this this signature to go searching. This is now single cell RNA sequencing from the human brain in individuals that died of severe COVID. And in this map, there are very clear cellular subsets showing up. Here is blazing. That again is microglia and not astrocytes. The astrocytes are here in red, which is here. And you can, I don't know if you can see, but in there, there are sprinkled some dark red dots, but not nearly with the expression levels or intensity that's expressed in microglia. And there's another little island of cells here in the middle, which is actually endothelial cells. So again, we got this LPS response signature from astrocytes. It's highly expressed in microglia, astrocytes, as well as endothelial cells in different contexts. Okay, I'm going to keep walking. Okay, so this is a fun example from Michael Kornberg's data. So he passed me this data in 2021, and actually very recently, I think within days, they just published the study looking at a bryostatin modulation of this LPS response. So they had, it says macrophages here, but it's actually a mixed glial culture that they used from mouse brain. And here, it looks like that LPS signature goes up in every LPS condition they have. This is LPS plus interferon gamma. This is LPS plus the bryostatin compound that has been shown to, in different contexts by Dr. Kornberg and others, to have anti-inflammatory effects in different contexts. And the last one is LPS, interferon gamma, and bryostatin all together. So any of the samples that saw LPS really turn this on, and there's a huge gap here. And I did not know that the paper came out until just last night. I emailed Michael and said, "Oh, can I show some of your data?" He said, "Sure, the paper's just out." So I took a peek at the paper, and this is a lesson in sort of data observation and data visualization. So it really looks like nothing is going on except anytime the cells see LPS, they turn on this signature. But if we just take one more step and zoom in on the data, so these are now 3 different LPS conditions: the LPS alone, the LPS interferon gamma combo, and then the LPS bryostatin. So very clearly, bryostatin does not bring things down back to control levels. There's a huge gap here. But on the scale of this induction, it does effectively reduce the total induction of this signature. So this is, one way of looking at how to use this LPS signature. Another in that, be careful how you look at data and how you plot data. You can miss things. And so I wanted to go one more step with this. I actually looked into some of the genes that they in the paper identify as interesting here. And this is the same list of 25 genes that we got from the LPS. Colored in red are genes in the top 5 or 10 that they saw different between their LPS treatment and bryostatin. So this LPS, the 2 different LPS experiments in different contexts already have a highly overlapping signature. Interestingly, one gene was actually in the opposite direction. And whenever I see a direction change, I wonder about mechanism and how that might be different across these 2 experiments. But in any case, this is trying to show you at the level of patterns where we have an LPS signature here, and it's shared here, modulated by a small molecule here. What are these individual genes? We can drill right down. And again, I don't know offhand which others on this list are involved in this signature, but we could go back through and look at this data one by one to check it out. Okay. This is one more. A single cell study in mouse spinal cord injury. When we project this LPS signature into that, there's some little pieces on this map. You can see there's little highlights here. This is the astrocytes. These down here are endothelial cells. And these, I believe, are the leptomeningeal cells down there. So it's hard to see on the UMAP, but if we just take another step, this is the exact same data broken into violin plots. So this is, again, the y-axis is now this same LPS signature. We have it broken down by cell type and also by time from injury. So the first violin is uninjured, and then it's 1 day, 1 week, 3 and 6 weeks. And so what you first notice actually is microglia are not the highest. Those leptomeningeal cells are a little higher in expression. But what you notice about every cell type is that the day 1 after injury, all these cell types, virtually every cell type measured in this single cell experiment, is turning on probably many more things, but at least this packet of 25 LPS responsive genes are coming up immediately, or at least within this 24-hour period of spinal cord injury. And again, as we would expect from innate immunity, these are in multiple cell types. Here it's even in more cell types than I would guess. Perhaps only neurons really aren't involved, although there might be a hint of it. We'd have to look into these distributions. But it's really widespread, and you can just see it. As long as you have the data, you can see it like this. And again, this is also a lesson in how to look at data. This picture was very hard to interpret. It's hard to see what's going on here. You can just see a little bit of action here, here, and here. But the same data redisplayed, bringing in important metadata, all of a sudden shows us that this packet of genes is super active immediately and turns off pretty quickly. By a week later, we're down to probably, it looks like across the board, almost control levels. Um, so it's an acute response, which we would guess from here in the original experiment too, we could see that. Different ways of seeing biology with the same signature. And again, all of these are screen captures. All of this can be done by pointing and clicking in the NEMO Analytics environment, because two things have been put there. One is the interesting related data sets, and the second are the algorithms to do these projections. And in this case, it's simply the algorithm is just add up the 25 genes. But as we'll see later, when we get into the more mathematically complex decompositions, the math to do these projections is quite involved. Okay, so here we go. This is a recent study just last year. It is in the peripheral immune system, but again, as I keep saying, the innate immune system has lessons to learn or lessons to teach for all cells. But in any case, this is the kind of study that's really becoming across fields very common now, which is kind of an atlasing study. So they sequenced 400,000 individual cells, the RNA from 400,000 cells out of mice. Now, they used about 300 mice. They captured cells from a particular lymph node that was easy to access. And before doing so, they gave these 300 mice either nothing or individual cytokines, and they gave them 86 different cytokines. So that's why we're up around 300 animals. I think they use 3 animals per cytokine. I think it's 24 hours after cytokine treatment. They then drain those lymph nodes, capture single cells, and sequence them. So from 300 animals with a range of 86 cytokines, we have all the immune cell types that are coming out of this lymph node treated with different cytokines. So that all of what I just said is contained in this map. And the biggest effects that you see in the map, and this is pretty much across the board with this kind of data, are related to cell type. So your monocytes are over here. Neutrophils are kind of a dense, compact cluster down here. And then green, all of these are different macrophage populations. So I'm highlighting the 3 sort of innate immune cell types. But of course, as you see here, there's a plethora of B and T cells in the lymphoid lineage as well as dendritic cells in the whole array of cell types that are found in the mouse lymph node. That's where this data is coming from. And again, in the context of all these different treatments. And on here, you can't see which cell is treated with which cytokine, but of course, that's data we have here. So as we go through and look at this, um, that'll come into play. Okay, so now this is again a redisplay of the same data in the UMAP here, but in a violin plot. So now we're looking the expression of these 25 LPS genes across cell types in the mouse. And you can see that the cell types I was pointing out, the innate immune cell types, are the ones that have highest expression of this signature. And we don't know offhand which cells treated with which cytokines are responsible for the increase in this. And that's what we'll be interested in next. So now I really want to transition to which mouse lymph node. I'd have to go back and check. So if we, I can quickly do this. Let me do this for you. We can do this fast. Let's find out. Or in fact, what I'll do is I'll put the PubMed ID in the chat, and if somebody wants to go look it up immediately, you can do that while I keep talking. And that's another thing. Whoops, I went forward a little bit here. Sorry. So right in Nemo, if you're looking at this data in Nemo, there's a button you can click on that will take you immediately to the publication. So the Nemo environment is not only trying to bring data to you, but it's almost also like a scientific notebook. If you collect a couple data sets, you can surf the papers as well as the data in sort of an integrated manner. So yeah, you can check which lymph node, and please let us know, jump in and let us know if you see which node it is, what difference that might make. I'm not a immune system aficionado, so I'd love to hear that. But what I'm going to do is walk a little bit more through this same experiment to now elevate the precision and power with which we can create these signatures. Or not create them, but identify them and then use them as molecular tools. I often call them molecular tweezers to go pick out particular parts of other data sets. So here actually, does it say, it doesn't say here, lymph nodes, it just says nodes here. So this is a clip from the paper. So they have, this is just their experimental design. And I sort of described all this to you already. But now I'm going to bring you to a little bit of the details of what we've developed in that biostatistics/mathematical collaboration. So once we had done a lot of decomposition of one data set and then gone and looked at those signatures in others, eventually you get to be like, well, I'm using these 3 or 5 data sets over and over. I'm decomposing one, looking at the other, then decomposing the other one, looking at a different one. Why not decompose them at the same time and just say, okay, I have 3 different challenge experiments, a cytokine challenge, an LPS challenge, and a something else challenge. And I want to know what they have in common. So that's how we designed these joint matrix decomposition tools, is that you give them a number of matrices, and you, by breaking up the matrices in particular ways, you make the algorithms focus on particular things. So in the context of this data set, you could think of the whole thing as one matrix where you have your 400,000 columns and 30,000 genes as your rows. The 400,000 columns are individual cells that span both cell types and cytokine treatments. But the way that the algorithms that we developed work is they first look inside a matrix for patterns. And if you gave it 3 matrices, it'll look inside each of those 3 before it compares them, inside of each one to see what's going on. And then it'll compare, we'll say, well, are the things happening inside of matrix 1 also happening in 2 or 3 and vice versa? So the algorithm can only see things that are inside one matrix. It never looks at the difference between matrices. It only looks at them inside. Now, why does that matter? The 2 bullet points here that I have listed are why it matters. If we split this data set up in 2 different ways and feed it through the algorithm, it will find fundamentally different effects. So if option 1 would be to split this giant data set into little matrices where each matrix is a cytokine treatment. So it has all of the cells of different cell types treated with interferon gamma in one matrix. And then all of the cells treated with TNF in another matrix. It will then look inside each of those matrices separately for differences in the data, and those, in this case, will be driven largely by cell type, because the biggest differences in the data set are by cell type, and the thing that changes or that doesn't change is the cytokine treatment. So it's lots of different cells being treated with the same cytokine. So the big difference inside that matrix is cell types. So if we fed it 3 different cytokine matrices with different cell types, all of the pieces you get out that are shared across those matrices are differences between B and T cells, or differences between your innate immune cells and your lymphoid cells. If you take the same data set, and instead of grouping it by cytokine treatment, you group it by cell type, and you say, for instance, give it 3 different cell types as 3 different matrices. Now, inside each one of those matrices, there's only one cell type, but there's lots of cytokine treatments. So the thing you'll find is common cytokine effects across cell type. It'll be totally blind to the differences between cell types. It'll only see cytokine responses. And if you have a common cytokine response across the different cell types you chose to put in, you'll find those. Okay. No details on the lymph node. There was a note in the chat. Okay, so this is now an example of me breaking up this data set just how I told you in the last example, where I'm going to give it 3 different matrices, each containing data from only one cell type. And I grabbed the 3 innate immune cell types, or the 3 sort of principal innate immune cell types that we're getting out of these lymph nodes: monocytes, macrophages, and neutrophils. And I showed you those here in the map. Monocytes are over here, neutrophils down here, and then all these green around the outside are different macrophage populations. So again, this is just saying exactly what I said leading in here. Because we chopped it up by cell type, what the algorithm is seeing then is the range of expression variation that comes with different cytokine treatments. So I did this decomposition. It's called a joint NMF, where NMF is a non-negative matrix factorization. This is some of this fancy math that my biostats collaborators helped me develop tools with. And we asked for 25 patterns, which means give us 25 cytokine-related expression changes that are shared across all 3 of these cell types. And this is one example. This is pattern 9 of 25. And before we jump to the live website, I want to show you the kind of pattern that shows up is a direct result of how we structured things. So if you remember, and at this point, I feel like I'm repeating it too much, but we're looking at effects inside each matrix first. So this bunch of monocytes, right, these purple guys are monocytes. That's one of the matrices we put in. So it's going to find differences across that. And you can see, I hope this isn't too small, that there's one end of the monocyte cloud that's really expressing this particular pattern, pattern 9, and the other half isn't. And that's actually across the board, almost any cell type you look at, but let's look at the ones we used first. Here are the neutrophils. And again, if this is too small, I have another graphic coming up that'll make it better. One half of this is expressing the signature and the other half isn't. And then in each of these macrophage clusters over here, you get the same thing. This is a cluster of macrophages where one corner of them are expressing this pattern 9 and the other ones aren't. And here's another macrophage cluster, same thing. Part of them is expressing it and part of that cluster is not. And even in cell types, now this is starting to get fun here, even in cell types that were not included in this decomposition, for instance, this brown cluster here is natural killer cells. There is one end of these natural killer cells that are expressing this signature. They're in some different state. And if you look around, these are the B cells. There's one corner that's expressing this. These are some particular kind of dendritic cell, and there's a little corner that's expressing this pattern 9. So it's a gene signature that is very prominent in the 3 innate immune cell types that we looked at. It's in a small but very distinct and highly expressing subpopulation. And it's something that's not limited to the innate immune system. Almost every other cell type we look at in these clouds has a small subpopulation overexpressing this pattern 9. Okay, so now I do want to jump to the website to do a little surfing, but I think I want to show a couple more pictures first. Let me see if I can remember my. Oh, this is what should have been showing you. I made it a little bigger, but again here, just to zoom in, that's the monocytes, high chunk there. Here's one macrophage cluster, the highly expressing subset here, and so on. You see these subpopulations in the larger clusters that keep coming up. Okay. And this again is because we designed this experiment from the beginning to look for things of this structure, things that are repeated molecular signatures across cell types. Okay. So I think I want to show you these pictures and then jump to the live website. This CXCL10 is a cytokine that is the single top gene in this pattern. So it's the biggest contributor to this pattern that is expressed in that whole range of subsets of different cell types. And here it's broken down by cell type, so we don't have to try to read the tea leaves of the UMAP. But as you can see, and now also we have which cytokine they've been treated with. So this is the monocyte cells. So that's these guys here in purple. And ignoring all the other cells, we're now looking at the monocytes broken apart by which cytokine they saw. So it's the interferons. This is many interferons. They've been grouped. And then the gamma gamma chain slash IL-13 slash TSLP cytokine. So these have been grouped. They did 86 different treatments. So each of these is kind of a related group of cytokines. But the cool thing is that now we can see not only that these are shared molecular signatures that happen across different immune cell types. Sorry, I'm reading the questions. You initially specified last time. Yeah, so I'll talk about the 25 patterns in just 1 second. Yeah. So, but in this one of those 25, we can now see that not only is it specific to particular cell types, but it's specific to particular cytokine treatments. And so now we have a cytokine specific, or at least a cytokine class specific signature that's coming from the innate immune system of the mouse, but clearly shared by some of the other cell types in the mouse as well. And now I'm going to jump to the live website. But before I do, the 25 patterns is indeed arbitrary. So when most of our algorithms, you can name the number of dimensions you want. And there's two ways of looking at that, the biological way and the mathematical way. The mathematical way in general is when people call it the rank or the dimensionality of a data set. So knowing what number of dimensions you should look at, in my head, you need to use both those perspectives, the biological and the mathematical. The mathematical one is to say, well, what if I ask for 1 to 100 dimensions and I look at how well the data is fit by those different, so you get a dimension that looks like this, where this would be, say, your error in fitting the data. This would be 1 dimension to 100. And you look at how much better you can explain the data as you add dimensions. And you look for things like an elbow. If people are aware of the scree plot that looks at PCA analyses, you end up getting what ends up being diminishing returns. You get lots and lots of error reduction every time you add a dimension up to a certain point, then it starts to flatten out. So that's a nice point to take. But biologically speaking, the other thing you need is just level of resolution. If you wanted just to see, if there were 3 cell types in your experiment, and you wanted basically to see the cell type effects, you can ask for 3 or 4 or 5 dimensions. And so that would be somewhere up here in the curve. But if you really want to get into the molecular specifics of things and break down what's happening inside of each one of those 3 cell types, you're going to need 10, 20, 30 dimensions, which is going to be up here. So in general, in the most involved cases, what I usually do is look for a scenario. I'm going to redraw this same thing here. And what usually happens is something like this, where I look for a low dimensional and a high dimensional level that gives me the most computational bang for the dimensional buck, and is at one at a low and one at a high resolution. Because there really is no perfect answer to this question of how many dimensions are there. In fact, even 30,000 dimensions, one for each gene, has been greatly reduced. We're totally ignoring splicing by doing gene level analysis. So, even looking at genes is sort of a dimension reduction step of the first order, going from hundreds of thousands of loci in the human genome that are expressed in differing ways, getting squished down to protein coding genes, and without splicing. So in any case, that may be a long-winded explanation of the dimensions, but the 25 is arbitrary except in the context of the ride of looking at these dimensions that I just described there. And I think it's important for both the biologists and mathematicians to give a little on that. And what I just said probably ruffles some mathematical feathers, but satisfies biological. But the mathematicians also have to realize that there isn't one level of analysis, even if it is most efficient or least error.
Can I ask a sort of clarifying question? This might be more involved.
Absolutely, jump in, Ed. Great.
So I understand the comparison you're trying to make with the scree plot in this instance. Are you also sort of generating a scree plot to then choose your dimension here, or is there a different type of plot that's being generated? Like
So
what
it's
exactly are
not
you
a scree plot just because it's calculating something slightly different. But yeah, I really like to have an idea of where I'm getting the bang for my buck and error. And in that, actually, the Sonthalia et al. Publication I mentioned at the beginning, I had made this, but actually hadn't shown it. And one of the reviewers asked about dimensionality, so it's going to go in the paper. And I do
Mhm.
exactly what I described. I pick a low and a high. I don't know what to call it because the scree plot is specifically for the percent variance in PCA
Right,
plots. But in
right.
this error plot, yeah, I usually really liked, and I usually pick a high and low because you want a bird's eye view of your data and you want some details from the data. And you can inform that with this kind of plot. Does
Okay.
that
And
answer
then
the
this,
question, Anna?
yeah, yeah, that did. And this might be jumping ahead, but when using Nemo, is that something that you can basically just look at it and choose, I want this high one,
No,
this
yeah. So the only thing you can't do in Nemo right now that we want to get in there is the decomposition that I'm talking about.
gotcha. Okay.
So what I can do though is take this decomposition I did offline and upload it to Nemo.
Uh-huh.
And in fact, if that's the end of your question, I'll jump to the live website now and show you exactly that. But
Yeah.
I'm glad you brought that up because it's the only missing link currently in doing everything I showed you online with points and clicks. So all the data is there, the surfing of it is there, and the looking at it. I did the decomposition of this study offline and then uploaded the gene list. So each of these 25 patterns has a gene list associated with it with a weight for each gene. And that's how we do the projection with all
Mm.
the matrix math in the background. And so let's jump in and look right at it. So if I click on this, and again, if you get these slides, you can do exactly that. If it's in presentation mode, you can click right on that. And this is actually going to take a second to load. So I am going to go, I have this open elsewhere already. So this is the Nemo website. So over here, there's a little sidebar with lots of things you can do. I'm not going to get too practical today. Absolutely reach out if you want to learn how to use this. Um, this is the data collection we're looking at. So each box here, just like in my talk, you can already see these kind of look like plots we've seen. Each box is a paper or a data set. Some papers have multiple data sets. This neuroinflammation collection has a huge number of data sets, as you can see here, all on this one page. And this is my long-winded way of saying, this is the last author of that study, the immune dictionary study we just looked at, where I grabbed the 3 innate immune cell types to look at cytokine effects. And I asked for 25 patterns with a joint NMF. So that's what these are right here. So I'm clicking on pattern 9, and I'm going to slide down here to where we started the talk. So now this is a view of that Ben Barres experiment that we grabbed an LPS signature from. And this turns out that this pattern 9 that is shared across the innate immune cells of the peripheral mouse lymph node is also slightly increased in astrocytes centrally. So, and again, we can look behind the scenes at which gene, or behind the picture at what genes this would be. But for now, I'm going to walk through a number of different data sets just to show you sort of the amount of transcriptional landscapes and experiments that we can look across with this. And just to show you a little bit of this, if you click on these 3 dots, this will take you to the PubMed entry for this data set. This has just a quick and dirty description of it. If I have any other way of displaying it in different ways, if you can create these custom, you can create different ways of looking at the same data. As I showed you, looking at different pictures of the data in different ways, you see totally different things. And the other thing I can do is say, I just want to look at this one data set right now, kind of clear my mind, make a little bigger plot, and there it is. Both in the LPS and the middle cerebral artery occlusion model, this innate immune system signature is present. And now just to look at sort of the diversity of data that we can get into here. This is another LPS experiment, but done in the context of Alzheimer's genetic engineering in mice, where we can see that not only is the LPS higher than the control, but the genetics itself actually give you an effect. Now, again, the y-axis here is our pattern 9 that we got out of the innate immune cells of the mouse. And we're now looking at, these are microglia sorted out of the brains of genetically engineered mice under LPS treatment. So again, just the data gives us the power, the data and the signature together, because I had to upload these signatures to Nemo. But for instance, to give you an idea, if you're reading a paper and table 2 is a list of genes that go up in a condition that's of super interest to you, you can download that table, upload the list of genes to Nemo, and any data set that's in Nemo, then you can look at the composite expression of that list of 20 genes from table 2. This is a little more fancy because we did that whole decomposition. But as far as what you can do if you don't code and you don't play with these giant data sets, even without coding, you could download a table, upload it to Nemo, and check out whatever it is you want to check out. Here's another one. This is a combination of LPS and cytokines in primary microglia. So, and again, we're seeing this effect. In each case, it's important to remember what we're coming back to. It's a new experiment each time, but it's the same innate immune response that we got with that joint decomposition. And this is one I really wanted to show today because I know of the interest of many in this audience. There is a dearth, a paucity of in vivo human RNA sequencing in the HIV literature. There's a couple places starting to produce it. But this is from 2012, so this is quite old data, but incredibly useful. They have controls that are HIV negative, and then 3 different groups of people living with HIV that had differing cognitive status. So the increasing red goes from no cognitive impairment in pink to some neurocognitive impairment in the red, and then that brick red is full-blown HIV-associated encephalitis. Now, this signature that we got from the innate immune cells in the periphery of a mouse is clearly associated with this worst case scenario. The encephalitis group in all cases, no matter if you're in white matter, cortex, or striatum, is really, really up there. Now, I've looked at this data set a lot, and there are not a lot of things that look like this particular pattern. And again, coming back to what we're looking at, it's important to remember this signature did not come from this data set. We got the signature, this pattern 9, from the innate immune cells in a mouse lymph node. But again, you need to, whatever biological context you want to explore with these data sets and tools, you really got to know the system, because if you didn't know about the innate immune system, it would just seem like everything is being expressed in the wrong cell types. None of the cell types we saw in the mouse lymph node exist here, except we know that microglia are the tissue resident macrophages of the brain. So this makes sense. And if we have single cell data, we can get into what exact cells we think are responsible for these changes. But this one was super exciting to me to see how clearly not only associated with HIV, because even in there is some difference between controls and non-cognitively impaired individuals. So this is always kind of brewing in HIV, but if you have encephalitis, this transcriptional program has sort of shot off the roof and really, really, really high. Okay. I know we're at the end of our time here. I'm going over. So I think what I'll do is actually go back to my slides, and I think I have one last image to show. This is just to show the same thing in macrophages there, so we can skip that. Actually, it's two last slides to wrap up. One is just to give you an idea of what's out there. We've been talking about these big data sets. This one had 100 or 400,000 data points in it. And just recently, this company released a 10 million cell experiment. They're just trying to show people what they can do, so they put it out there for free. From 12 donors, they do 90 cytokine treatments and 20,000 perturbations, that's sort of cytokines by cell types. So there's sort of an in vivo human paralog of this study, and you can just start to imagine the different ways we could chop up either one or both of these data sets into submatrices to focus questions and get signatures out. And similarly, centrally, there are huge numbers of data sets and atlases coming out to look at astrocyte and immune responses this way. So the data and resources are only growing to do more of this. And I'll end with just looking at the expression of a single gene. So if all of this is just a little too complicated or some of it is too far out there, what can Nemo Analytics do if you just click on the front door and say, I want to know about the, what is the name of this gene? It's folate, what is it? It'll come up here. But this is the gene actually that Dr. Barbara Schlusser sent me because I asked, well, what kind of genes are people interested in? I want to show something of interest. So this isn't a whole story, but this is just to say, even if you don't have some crazy decomposition or experiment done, you just have a gene of interest and you want to look at it across data sets that you're interested in, this folate hydrolase 1 gene is expressed here, which is the oligos in this data set. This is single cell sequencing from the human cerebellum. If we keep going down here, there's tons of data sets. Here again is oligos. This is the COVID data set. Again, brain cells in COVID. The oligos are expressing most of this, but it is sprinkled in here in this neuronal cell type. They come down here. In the sorted cells, it actually looks like it's in astrocytes more than oligos. So this also kind of gives you a picture. Well, not everything is always consistent across experiments. This was kind of surprising to me, is that the single cells looked like it was in oligos. The sorted cells looked like it was higher in astrocytes. Here, it's pretty well expressed in the astrocytes in this Ben Barres experiment that we've been looking at. And this is one now, and I'll end here and take questions. This is now mouse data in a spinal cord injury model. And if you notice, there's this group, which is what we've been seeing in the other single cell data in human, that it's expressed in oligos, but these are astrocytes here. So it's also expressed at high levels in astrocytes. I don't know if that's a difference between those two single cell experiments, or if the other one, it's because it was human and human astrocytes perhaps express less. We'd have to look systematically across more studies. But the idea is simply that it's out there. We can find the answers to these questions. So if you're studying a gene and you don't do any multi-omics, you can come look up your gene and just see where it is and what conditions and cell types it changes with. So I think I'll stop there. And I know I've gone over time. I apologize. But please do reach out, questions now or via email or any other way to see how I can help you use this in your own work.
Thank you so much, Carlo, for a fantastic presentation and sharing your passion with us in the world with these resources. For those that are still on, if you're able, if Caroline, we're okay, we can stay on. And yes, go ahead and ask your questions. Just unmute and ask your questions.
Yeah.
Carla, this is Barb Slusher.
Hi.
Since you just showed full H1, which I asked about, that thank you very much for looking into that. Just a question. So nobody's ever described this protein in oligos? It's been well described in astrocytes. So how do you take that differential information in the single cell? Initially you saw it in oligos and then ultimately in astrocytes? What do you
So,
conclude?
so yeah, I mean, you sort of instantly saw how this can get messy, right? When you see everything, it's not always a great, easy picture that fits together. So let me repeat what you said to make sure I heard correctly, that the protein level of this gene has been characterized in oligos, but not astrocytes, correct?
Opposite.
Opposite. So it's been characterized in astrocytes, but not oligos.
Correct.
Yeah, that to me is amazing, given those first couple plots, right? I mean, in fact, let's even take it. So that is, for instance, here, right? So,
Yeah.
and this is actually work I was involved in. This is the oligos. The astrocytes are right next door, but they do not look at all like they're expressing it. Now, of course, we're looking at RNA here, and
Yeah.
I know that's a little small. You can always pop out the image and
Yeah.
make it a little bigger. So, but really, it's not there. So, as I said, though, when we looked at the sorted data, it is there,
Right. And
and
it
this is
says.
part of getting into the weeds. Yeah, the sorted data here. This is mouse data now, and it looks way higher. And these are log units over here, so it doesn't
Yeah.
look like an enormous difference between oligos and astrocytes. But I rarely see a cell type discrepancy like this.
Yeah.
This is sort of a class, and there's two of them, right? Here's a second brain study. It is blazing here. And
Yeah.
astrocytes are in red. There's very little expression in the astrocytes. So I don't know if they produce the protein and it's stable, and they don't need a whole lot of RNA around to keep producing it, or what is going on.
Yeah.
But there's some disconnect because it's a really big difference even across studies. And before we look at protein data that you're talking about, this is all RNA and it's not even consistent here. Some of these studies really show it higher in the oligos, some show it really higher in the astrocytes. I found this was interesting. This is yet another Ben Barres study, just to give credit where credit is due. So many useful studies that he provided us. In any case, the highest samples are these sclerotic astrocytes, but then all these other mature astrocytic and fetal astrocytic samples do not show this gene. So it might even be a context specific
Right.
scenario where things are turned on. Um, so I'd be very happy to sit with you and give you some of these links and surf some data with you to see if we can get any more clarity. But this is a good example for showing what usually doesn't happen, actually. It's a good example to get you into looking at an individual gene across many data sets, but it's usually the conditions that get murky, not cell types. Cell types are usually pretty consistent. So I'd be interested to see what this story is actually telling us and why these different cell types and different experiments are showing really different levels of this gene.
Well, thank you. And I'll follow up with you after. I'll follow up with you after.
Great. Yeah, I'd love to talk with you.
Hey, Carlo, can I ask a question?
Yes.
Yeah, so I'm sorry I joined late, like 20 minutes late, and missed the important talks, your talk. It's very, very informative to me because I'm new to the field. So I'm working on like innate immune cells. So in the brain, what we should focus on, like based on your data, like astrocytes or microglia, something special, because I'm working on neuroinflammation in vivo, all the in vivo, not LPS induced anything, we HIV model. So which
Okay.
we should focus, because I remember you one slide you saw CXCL10 one chemokines, they significantly increase variation. So we should focus on the astrocyte or microglia or some other cell types.
So I don't think my current work would direct you to look at any one cell type over another. I think what my my story would direct you to do is step one is take the genes or cell processes you're already interested in if you have some realm of interest, and whether it's in your own data or in these data sets, just like we were looking at the folate hydrolase gene, look at some hooks, some either signatures or individual genes you are interested in, see where they are expressed, and know what cell types to go into. Because one thing that I have learned in my little time in the innate immune system is that cell types can surprise you. It's very common that if I get an activation signature from a single cell type, that it shows up very strongly in endothelial, microglia, astrocytes, even neutrophils or macrophages that are coming in from the periphery. So without any other information, I wouldn't say there's some key cell type to focus in on here. I think knowing what it might be more interesting actually is to know some molecular pathways
Okay.
that are shared across cell types, but that can be therapeutic targets across indications. I think that's kind of the mode we should be thinking.
Okay.
Yeah.
Okay, thank you.
Yeah, you're very welcome.
Well, I think the rest of the people in attendance or have questions will have to follow up with you individually, Carlo.
Great,
Again,
please
this was
do.
fantastic. And I know you see, everybody knows, see, I have a lot of homework to do. Thank you so much.
Great talking with you.
Great job, Carlos. Fascinating. Thank you.
Thanks.
Thanks, everybody. I'll send the announcement with next week's speaker on Monday. Thank you.
Thanks.
Thanks.